Hello and good afternoon, everyone. Good morning, everyone. Um, I think it's time for us to start. Maybe if others will join in the meantime, they can connect. So I would like to check if Dr. Nasim is here, as he is the star of today's
Hi,
event.
Thomas. Good morning. Yes, I'm available.
Hi. Okay. Um, so this is the 14th CarboHyde Masterclass, and unusually to the former masterclasses, this one will not be about psychodextrins. So as you may know, or you may not know, CarboHyde has a branch in India, which is led by Dr. Naseem Khan. And in this Indian company, we don't only work with psychodextrins, but we also work with other products like silicon dioxide. And we dedicated today's session to go a little bit deeper into this product, what is possible with them, how can it be used in the pharmaceutical industry, what kind of innovations we are working on, what kind of projects it can be used. And this will be introduced to you by Dr. Nasim. As you can see here, I've known Dr. Nasim for several years. I think it's close to 10, maybe. We started, of course, working together on psychodextrins, but later became really good friends. And Dr. Nassim holds a PhD in genetics and has a very long and versatile background, both in research and in the business side of pharmaceutics. So it's my real pleasure to work with you, Nassim, and I really look forward to hear what you got to say to us about our silicas. So I think I'll allow you to share the screen. And then, okay, this is not the right button. And then, yeah, there you go. So you should be able to share now, and then the screen will be yours. As for the audience, the setup of the masterclass will be identical to former ones, which means that we'll first hear what Dr. Nassim has to say about the product. So it will be a short technical presentation of about 30 to 40 minutes. And then you will have the chance to ask questions. You can drop the questions in the chat box during the lecture if something comes to your mind, or you can just ask it afterwards as you prefer. The whole session will close at, okay, depending on when you're in 1 hour. So it's 55 minutes from now. So, Nassim, please start. The stage is yours.
Yeah, thank you. Thank you so much, Thomas, for this introduction. And definitely, I'm going to introduce myself. My background is not from pharmaceuticals. I'm from, biological science background. Uh, I'm engineering genetics. And after that, yes, business development. And now associated with CarboHyde, and we are doing a lot of research and yes, cyclodextrins. And apart from today's talk is how silicon dioxide is playing very important role as an excipient, and it it really helps how you can stabilize bioavailability, and many more things can be done with the help of silicon dioxide. So here how it works, I'm going to share my screen here. Yeah.
Okay, it works. We can see. But you muted yourself, Nassim. You're still on mute, Nassim. We cannot hear you.
I think it's audible now.
Yes, it's good now.
Okay, thank you. So, thank you, Thomas, for this session. And we have really worked hard from last 2 to 3 years. I'm going to share all the stories, and definitely we are going to discuss how silica plays very important role in the formulations. So here, how carbohydrate came into the picture when we have met. Uh, when we have met last, we know each other from last 10 years, as you said, but from last 3 years we have discussed a lot, not only cyclodextrins, yes, of course of silica. So we have came with mesoporous silica, and as to introduce, this is silicon dioxide. We are going to talk silicon dioxide in pharmaceuticals and how it is very important as an excipient in the future formulation, especially for dry syrups and tablet section. So we have, as a carbohydrate, we have a series of mesoporous silica, that is the oxide tab, OL, and BR. There we have three grades. So we are going to talk step by step that. In the coming series also, we are going to dig more and more about silicon dioxide. So here I'm going to introduce how silicon dioxide plays an important role. This is a reference, like how important is, and we have taken all the literatures and references here to show the audience. Yeah, yeah. Understanding what is porous material. Porous material having three categories: uh, microporous, mesoporous, and macroporous. So here is the porous diameter. It is like it is less than 2 nanometers, high selectivity, and then mesoporous come into the mid between 2 to 50 nanometers, and then the porous is on the higher side, like 50 nanometers. So we work on mesoporous. Uh, the reason uh, here why we are working mesoporous, because selectivity and how the behavior, and we can the canvas is big when we talk about mesoporous. Yeah, we can talk about microporous and macroporous as well in the coming time, but today is mesoporous. uh, it is how differentiate in the coming slides. I'm going to share how important is and how it is beneficial for the formulation. Here, the mesoporous silica are versatile material. The morphology, here you can see a structure that it seems like it's a golf ball, which is having a lot of pores into that. So when we talk about uh, this golf ball, and when we talk about silica, silica is generally used for moisture protection, and most of the formulators which are present in there, they use silicon dioxide mostly to protect their formulations from water content, from the moisture, to have a better stability. Here, uh, here you can see, if my cursor is visible to you, like your API goes and trap here. It's sometimes it's API, sometimes it's water content, sometimes it's moisture. So it depends on formulation to formulation. So how the morphology is like highly ordered pore structure. It's maybe it's random also. So this is the difference between sometimes when we, as a terminology, when we talk about like mesoporous or it's only porous. So when we talk about mesoporous, it's sometimes it is like it is ordered very highly precise, so that you can have a better result in that. So now dimension is tunable pores. So when we talk about mesoporous silica, it is very much tunable, and you are getting better results because of identical and the control ranking from 2 to 100 2 to 10 nanometers, allowing it's like tailored molecule which we manufacture. So exponential surface area, so providing massive internal surface area for high capacity interactions and reactions when it interacts with API. So in the coming slide we have the differentiations, like how many APIs, how with many type of silicas as well, how that behave, and how what is the benefits between mesoporous and other silicas as well. So here's a small morphology, how this is a picture that we have taken from internet. And uh, we have our own pictures, but it was not so much clear and visible. So we have I have taken a picture from the internet. So this is how mesoporous silica morphology looks, and this is how the pores look when we see in the microscopes. So here you can see it is like 20 nanometers, 1 nanometers. Here you can see 50 nanometers. This is a how mesoporous silica looks like. Yeah, this is very important. And uh, this is important because silica, and people think about what exactly the differentiations, what exactly how you differentiate the different type of silicas, you know. So you can see here, like this is a difference between fume silica, then silica gel, then spherical silica. There are basically, if I'll talk about, there are four type of silicas. uh, it's like precipitated silica, fume silica, then mesoporous silica. Sometime in the category of mesoporous, microporous, all comes under that. Then there is a type of silica that is called hollow silica as well. So majorly there are four types silica. So conventionally, people from long time they're using fume silica because it's easy to process, easy to manufacture, and if we talk about like let's start with fume silica, because most of the formulators present here, they have used fume silica. You can see here like some brands like Aerosol, Cabosol. These are the brands you people maybe you have already used. The fume silica is something like flex, there's a flex, and it is it if you see here uh, on the picture, it is something like flex. So the moisture uh, terminology, if we talk about, so there are two terminologies. One is adsorption, one is absorption, AD and AB. So here, when you talk about fume silica, it only adsorbs. That means this your water content, your moisture stacks on the surface of silica. So the countries which is having very high temperature or very diverse temperature, like maybe Africa, India, maybe the Gulf side. So if your formulation is going there, so maybe you will face a problem because of temperature, because your moisture content is on the surface, it is stuck on the surface. So when heat generates too much, maybe if it is touching more than 45, if we talk about like Indian context, like we have a temperature of in range of minus 40 to 45, it's more than, sometime it's 50. So it is like the stability and correlation between stability and you know and the formulation, the real formulation which is present, your tablets, your dry syrup which is present in the market, and when they are absorbing the heat of 50 degrees or more than 50 degrees, so it starts degrading because of the selection of the silica, because you have selected some silica which is not suitable for such a high temperature. You need a silica, you need some excipient which even sustain in the high temperature as well. And that is something we are going to discuss, and that can be, your fume silica can be work better in related to fume silica, the mesoporous silica can be work better. This is a generic name of silica. Silica gel is a very generic name. So I have put in here because of just to understand better as a generic name. So we have silicon dioxide, tab, oil, and B. Other three grades, and three grades have a different porosity. This is the only thing, and different porosity shows how. I mean, sometimes you need different porosity for tablets, different porosity for dry syrups, or different porosity for maybe some other formulation. So this is we have gone with different porosities just to understand different formulations. Yeah. So here, when we talk about mesoporous silica, it is something like this ball. It's like golf ball, and if it is, it's just symbolic, just to show you people that it is something like ball, and it's having pores. So when your formulation is in the high temperature, actually this type of silica.
ఎక్కితే తిన్నగా వైజాగ్ బైపాస్ అయిపోయి వెళ్ళిపోతావు. బైపాస్ ఇది.
Hello. Yeah. Some voice is coming. Okay, anyway, so this type of silica, it's absorbs. That means there is a word called AB, absorb. So it goes into the cavity, it goes inside the silicon dioxide, and it traps. So here we have one silicon dioxide oil grade that we call as C oxide oil. Here, when we convert from oil to powder, so we have seen the efficiency of 75% of oil can be converted into powder. And if you if you punch also in the under the punch, when the high density punch making tablets, the oil is not coming out. That means it is not spilling out as compared to fume silica or the precipitated silica, because it is trapped into the pores. So this is how more beneficial when we talk about uh, problem wise. Maybe some formulators have seen the burgeoning problem, or some air is coming into the blisters of the tablet. This is also one of the problem of silica, because if the heat is more, the moisture comes out. That means on the moisture is on the surface, and when it comes out, it is creating a bubble type of it shows like some bubble is coming from the blister. So this is also one of the thing what we have experienced when we have did some local research, when we have did this type of research in where very high temperature is there. So this is sort of problems that we have sorted out with the help of our silica, that means carbohyde silica. Okay, so now the functional description, like uh, how it works, like it is like molecular trapping. So it traps, and the host, it traps in your specific molecule. The host is an active ingredient, and you can transport it. This is what I have shown you in
So,
the previous slide, that you can convert oil also into the water, or any API into
Hi,
powder.
Nasim sir. Sorry to interrupt you. I have a one question.
Okay.
Uh, so you have explained the different types of silica. So in that we are focusing on the mesoporous silica. So the size you mentioned, it is around 2 to 50 nanometer, right? So may I
Yeah.
know what exactly the strategy you are maintaining this size with batch to batch? So is there any variation, and what exactly the strategy to maintain the porous size? And if is there any variation, how it will impact on the product performance? Along with this, is this silica is generally dedicated for the oral product only, or we can use this for the parental product as well?
No, silica is for orals only, not for parentals,
Mm-hmm.
because it is not,
Okay.
it cannot be dissolved completely
Mm-hmm.
into the water or any media. It is just
Mm-hmm.
dispersed. So
Okay.
it is dispersible. I mean, you cannot filter it properly from the metabolism.
Mhm.
Yeah, of course, we
Okay.
are maintaining this pore size with our on the factory, and yes,
Mm.
if the pores are differentiated, uh, in the previous slides I have shown there are different pore size. If you can see here, yeah, this is a micro, meso, and macro. So if the pores is differentiated, yes, of course, the impact is different. So the mesoporous is something mostly used in the formulation, because it is like, if you talk about, if you know cyclodextrins like alpha, beta, gamma, so that is also totally depends on active, what the molecular size it is. So all the mostly these actives are come under the range of this mesoporous, and most our main objective is to protect our formulations from moisture. So 99% uh, people use this type of silica to protect from moisture, and there are many more advantages people are using. But if you talk about like tablet or dry syrup, our main objective is to protect from moisture. So this is the reason this tunable micro size, I mean the size of the porous pore of diameter, if you talk about, so this is the reason we have put in from 2 to 50.
Okay. Uh, Thank you. So, okay, I will ask the question later. Please
Okay.
carry on.
Yeah. So yeah, this is this silica, how the host gas relationship happens. So this is silica, C oxide, and this is a drug product and release of the API. So as I just said, that it is dispersed. It is not 100% water soluble. You can disperse into the water, or whatever the media you are looking for. But yes, if you talk about to to have your API to convert into the powder, yes, 75% we have seen that can be converted from liquid to powder, or oil to powder, whatever you can say. But that totally depends what is the viscosity of the oil or the API, what you are going to convert from liquid to powder. So there are many more applications. I have put in few applications in the slide because on the coming master classes we are going to discuss more and more. But as a catalyst, as a drug delivery, so it plays very important role in the drug delivery as well, because it can carry your drug from I mean, just what we have discussed, like ideal for liquid APIs. Controlled release can be done with the help of mesoporous silica to improve, the stability and the bioavailability, because it disperses easily. So in the literatures, it has been seen that yes, the bioavailability part has been increased. So in the many literatures, you can cross check, you can see how mesoporous silica is increasing the bioavailability as well. Yeah, I mean, like uh, how the revolution and the pharma, the the liquid part, what we are converting into the powder form. So this is the core function what we have just discussed. So this is other very important thing is that here nothing very crucial type of process required to convert from oil to powder. It is just simple mixing, and you will get the free flowing powder. So I mean, I can, if you can please mail me, I can share you how in the lab also how we have converted from oil to powder. We have taken samples like peppermint oil. We have taken samples of cinnamon oil. So we have this type of already we have processed in the sample. We have got a yield of 75% of conversion. So this is, I mean, here if you talk about like other uh, maybe in the market you can see many more things that is used to convert from oil to powder, but the process is bit tedious. So there it is simple mixing, and you will get free flowing powder. So it's very easy. So it totally depends on expectation, the absorption capacity. It's more than 100%. than or equal to 50%. Totally depends on the viscosity. If it is like a very thick oil, so you'll get more less yield. If it is very thin oil or the API, so you'll get more and more if but generally if we the oils like mustard oil, so which is very thick, we have seen the yielding of 35 to 45% that we have converted from this oil to powder, since mustard oil is very thick. So that I have seen in my own lab. So this is essential oils, low melting, like waxes, then oils, vitamin E, K. We have already converted, then aqueous solutions and pal. So this is what we have already seen that we have converted from. So this is the efficiency how this our silicon dioxide is used. Yeah. Then yes, moisture. This is a very important part. So generally and mostly in amoxicillin and clavinate potassium, it is used. And if you talk about like this business development part, so most of the business we are getting from this formulation only. So this is the formulation. This is the API which is very much highly hydroscopic, and we are protecting this API, and we are already getting successful stories in this APIs when we combine our silicon dioxide. Regulatory compliance wise, it is already GRAS, so uh, it is safe. So uh, what we are following is an pharmacopoeia status. So it is already we are giving like USP, EP already there because we are an European company, and we are qualifying even Chinese pharmacopoeia also, since we can see China market is growing very fast. So it is quite pharmacopoeia status is quite safe. So now we are in the process of USDMF as well. So maybe in the coming time, we you can see us on the list of USDMF. Yeah, in the coming time, we are going to have a classes of in the depth. We want to discuss more how the liquid drug can be converted into powder, then solubility enhancement, then in the nutraceuticals it plays very important role, and the stability and the regulatory aspect, the future applications and advanced drug delivery, and in the nano medicines and very advanced drugs, and even the filtration of many proteomics and genomics, even T cells. This silicon dioxide plays very important role. In the coming time, the you can see more and more, you know, uh, very vast applications of silicon dioxide in the coming time. So in the coming time, we are going to discuss more and more, and definitely we are going to improve more, and we require more and more your as a customer, as an business partners. We want more and more clarifications and questions and guidance as well from your people, that how we can improve more. If you have some problems in the formulation, so we can discuss, we can help you out. And if you have some thoughts, so you people are always welcome on that. So this is all about today's masterclass. So in the coming time, we are going to discuss more. So this is all from today's time. So yes, any questions, please?
Yes, sir. I have one question. So we understand more about that silica and all the application of it. Can you please focus on the cyclodextrin related part? Means if you have, or is there any planning in the future, or do you have any portfolio which is more focusing on the cyclodextrin? Because currently we are facing some challenge to control the impurity. We have used the cyclodextrin, but with respect to the time, we have seen some description failure. We observe the haziness in the solution. So uh, different lots of the cyclodextrin when we use. So each and every lot showing the different description. And so, can you please focus on that part if you have any point or comment? Have
You
you
talk,
seen any? Have you
you're talking
observed
about
any problem?
impurities
No,
part.
no.
Will
About the description. About the description, because is there any contribution from the degree of substitution? Is there any contribution from the impurity what the cyclodextrin present? Because a lot to lot we have getting the different results.
Which
for the cyclodextrins, we have one more expert with us, Dr.
which
Thomas.
cyclodextrins are you talking about specifically? Which type?
Sulfur butyl
Okay. And you said that the impurity profile is not okay.
that are okay, but description wise it's somewhat not meeting the expectation.
What do you mean description wise on the COA? You don't have enough data or
No, no, no. COI we have the data, but with respect to the time, let's say 8 months or 6 months, we are getting the different description. But when you use the another lot of that particular cyclodextrin, in that case we are not seeing that. So what exactly the source of that different in this description of that particular cyclodextrin? What exactly the contributing factor?
to be honest, I don't understand the question. Do, do you know, Sim?
Or maybe we will connect later on this
Okay,
part.
you can email us about it. What I can say about the sulfabutyl, it has uh, there are some very good suppliers globally and technologies, and it can be produced very reliably. So batch to batch consistency shouldn't be
Hmm.
an issue. If it is, then maybe you should switch the supplier. There are a few options that you can pick from.
Oh,
Yeah. Any other questions related to silica?
Yes, sir. Nasim sir, one question for the moisture protection for the amoxy and potassium club, what you have said it.
Yeah.
Uh, Already in the market, the grace materials are there in the grade of AL1FP, I hope.
Yeah.
In which way your silica is uh, more perfect or more suitable than that AL1FP grade?
Uh, I mean, I can't say it's more perfect or more suitable. Um, it is it is almost same, but in LOD plays very important role when you select the silica. So the loss of drying what we have managed in our silica is less than zero point 0.4. In the COA, you can see we have managed to, because the less water content the silica has, it is going to perform more efficiently. So what we have, the LOD part of our silica plays very important role, because the LOD we have very lowest part. I mean, we have control zero point 0.4. So this is only the advantage. Otherwise, it is almost same.
Okay, fine. Thank you.
I have a question as well, Nasim. So is mesoporous silica only used for small molecules, or are there possibilities to use it in biologics as well, theoretically at least?
Biologicals, it is going to use only in the filtration part. uh, I mean, it is filtered. Yes, it is used to for the filtration, but it is not going to use as an to consume or something else, but for the filtration only it is going to use. In many silicas, that totally depends on pore size. Yes, people are using to filter RNAs or mRNAs or tRNAs. So there are advantages. This is advantage of biological sciences here.
Mm-hmm. And so how reproducible is the drug loading at the commercial scale to the silica? Is it providing always the same results, or how is it working?
Yeah, what we have seen in mesoporous silica, because we are using mesoporous silica. If you talk about like many more other options are available in the market, but mesoporous is consistent. So 99% yes, it is consistent, and we are getting almost same results. Variable, it is not more than 2 to 3%, sometimes 4%. Yeah, it depends on the size of the batch. If it is big size, sometimes it is used in API production or something like that. It is used in the APIs also. So variation is not much, but mesoporous plays important role because mesoporous means consistency.
Sir Nasim sir, you have said around three grades of cyanide, right?
Yeah.
And what is the main difference between these three grades, and which grade is useful for the oil loading and which grade is useful for the moisture protection? Can I have?
Uh, yeah, we have you have given a reference of this grace, and if you talk about like AL1 FP or 244 FP, so if you compare like our silica, it is similar. Our C oxide tab is equivalent to AL1 FP, and C oxide OL is equivalent to 244 FP. The BR grade is having lower porosity, and generally this grade is we are pushing into the nutraceuticals. But if your objective is to convert from oil to powder, then BRC oxide oil is best suitable, because you will get the, if you talk about a reference of peppermint oil, so peppermint oil we have converted 75%. That means you can convert 75% of your oil into powder. So the best for the tablets, best is sea oxide oil, and for dry syrups, the best what we suggest is sea oxide tab. And to convert from oil to powder, the sea oxide oil is best, what what we are suggesting. Still people use in their own formulations sometimes. If you take about a reference of alchem, this product is already commercial, so they are using sea oxide tab in the tablet, but totally depends on scientist to scientist what they take and how they pick up the material. So sometimes we suggest, but scientist decides what they want.
Yeah, and another small question, sir. If we dissolve our APA, which is oil soluble APA, in the Getafos oils, right?
Yeah,
And the same oil can be loaded onto your cyanide.
yeah. Getaphosphate we are already doing many things. So maybe in coming couple of months, you will come to know that Getaphosphate will suggest our silicon dioxide. So we have many rounds of meeting with them.
Have you done the release part from that? Once the APA, which is dissolved in the oil, is went into the pores of the your silica,
Yeah.
and it is if it is an example of immediate release APA, whether it is at the end of the pharmacopoeial limits, it is releasing perfectly into the USP media or not. Have you done any studies that?
So we have did some different studies, but totally depends on oil viscosity. Again, because different oil has different release pattern.
Right, right. Okay, sir. Fine. Thank you.
Hi, Nick.
Yeah.
Yeah.
Anybody else?
Yes, sir. About just cyclodextrin SME, I have a question for him. So, if the innovator product does not contain the cyclodextrin, and if the test product, which are we have adding the cyclodextrin into it, so what are the PK studies are you suggesting in that case?
What kind of administration route are we talking about?
Intravenous.
Okay, so we have experience in that field, and there is a very high likelihood that you will find no difference, and we even have experience with the with FDA consultations about such issues. So usually in vitro studies, we're sufficient for us. What we had to prove is technically we did a head-to-head comparison between the two formulations for the release of active ingredient, protein binding, permeation through membrane, and then the similar sets of studies. Also, they welcomed if the cyclodextrin complex was well characterized, so it was shown that it's not a very strong complex, how it's fitting the active ingredient into the cyclodextrin. So it was a set of in vitro analytical results, mainly kinetical studies about how the complex is falling apart, and followed by membrane permeation and protein binding studies that all showed characteristics within error between the two formulations. So that was sufficient historically. It's just a few weeks of work, and we were not asked to do any kind of even animal studies, let alone human studies at that point. And we did it for multiple active ingredients, actually. So I'm pretty confident that this will be acceptable.
Okay, so these many study we have to do like the protein binding permeation because there is a membrane
Yeah.
binding study. So all the study we have to do. We cannot skip anyone. Or if let's say our molecule is the hydrophilic, just for to control the some degradation product, we use the cyclodextrin. In that case, there is no any formation of the complex. But when there is just only weak binding with the molecules and the cyclodextrin. But when it is entered into the body, so is there any case or is there any analytical techniques to prove that our API and the cyclodextrin having a not a strong complex formation?
Yes,
So
yes. So technic
what are
that
the
that is,
technique?
that is exactly the information that you have to prove that the complex is not strong, because if it were strong, it would mean that the cyclodextrin affects the pharmacokinetics of the compound. So the well, it depends on the active ingredient, but basically any kind of analytical technique is capable of determining binding constant. We usually use isothermal titration calorimetry, but you can use NMR, capillary electrophoresis, even phase solubility studies. So maybe you already have the information to make that calculation um, from the development reports. But it's a very simple study to determine binding affinity, and then you can show that okay, it's indeed a weak binding.
Mhm. Okay.
Nassim, getting back to silica, I got a curiosity. So, is there any kind of literature or report where, because you mentioned that, okay, this is a good technique to improve oral bioavailability, is there any kind of literature that compares this, I don't know, to amorphous solid dispersion or lipid systems? Or if not, what would you say are the advantages of the silica over other type of bioavailability increasing techniques? You're muted.
Yeah. Again, it's totally depends on API to API. Uh, I mean, if you talk about like class 4 drugs, so mostly cyclodextrins are the best uh, used for the class 4 drugs. So there are literatures, yes, available that shows the improvement of the bioavailability. So it again, it's totally depends on API to API and lipid to lipid, which type of it is. So it is not 100% universal that we can say that in all the subjects or all what we are going to take, it will increase the availability. It depends on that. So uh, again, other thing is that, like, if you talk about comparison of others, especially for the cyclodextrins, it is 100% water soluble, but silica is not soluble. So it is generally used in the oral solids as well. But if you talk about like cyclodextrins, it is used in injectable grades as well. So again, it comes with how the formulation is, what type of formulation, and what your challenges you are getting. But uh, rather than bioavailability, silica is mostly used to protect. It is mostly used to stable your formulation, and to decrease the, the biodegradability, and to protect your formulation with the moisture. So this is a major objective. Apart from that, the literature has been found that yes, the bioavailability can be increased with the help of silicon dioxide, because it disperses the formulation.
Naseem sir, a small question.
Yeah.
You are saying for dry syrup, your product is very less, LOD value is very less, like that you are saying.
Yeah.
You have done any stability analysis for like a highly moisture APIs like amoxicillin, like that?
Yes, we have already done.
Okay. Is
Yeah,
better
we
than is better
and
than our competitors, or
competitor wise, we are giving stability of 3 years, and competitor wise, they are giving the stability of 2 years.
okay, okay,
So we are giving better stability. We are giving 1 year, 1 year, plus the stability part.
okay, okay.
Anybody else?
Yes, Thomas, sorry, one more question. Thomas, are you aware about that any third party available to provide the testing facility to prove the bioequivalency of the test formulation?
Yeah, we at, at CarboHyde can do that. But basically, I would think that, okay, this is a analytically, it's we are talking about a wide range of studies, but not super complicated experiments. On the other hand, of course, it's you have to have some experience to put together the proper list and execute the tests as they should be. So if I were you, I would pick psychodextrin experts like us. But basically, if you combine the list, most of the analytical CDMOs would be um, technically capable of doing the experiments. So what we are talking about, NMR studies, ITC studies, um, uh, dissociation, kinetics, protein bindings. Most analytical companies should be capable of these things. We can do it also as well, but technically, if you have uh, uh, if you put together a good request for analytical companies, they would be probably able to help you as well.
Okay. And in addition to this, have you seen any observation for respected to the cyclodextrin? If we increase the concentration of that cyclodextrin at particularly at a room temperature, have you seen failure in the description?
Have I
With
seen what?
with
I'm sorry, I didn't understand what.
Okay, I will repeat. If in the test formulation, if we are increasing the concentration of cyclodextrin, let's say from the 10 mg I went to the 50 mg.
Okay.
So by with the respecting increasing that concentration, have you seen the failure in the observation means like haziness or any particular
Oh,
in
um, so
particularly
you,
for the cyclodextrin?
yeah. So you shouldn't, but depending on supplier, I have seen it happening, to be honest. So basically, the cyclodextrin solution should be clear, and it shouldn't be hazy, even at higher concentrations. But in my experience, due to the different manufacturing procedures, it does happen that that it fails the test, especially as you increase the concentration. So again, probably it's a workup issue how the cyclodextrin is
Right.
produced.
And the interesting part is that we have seen this observation particularly at room temperature, not at 228 and 40 degree.
Mm-hmm.
So what exactly the contributing factor means with respect to it is the whatever that haziness, and haziness is not so much like if we look so carefully then and then only we can able to see that slightly haziness, and even we cannot able to separate it as well. And I think it's temperature dependent. Okay, thanks for your clarification on this.
Okay.
I have got a question, by the way. My question is, the we are talking about the spray drying about the silica. Well, what is the difference between encapsulation and this drying processes differences for flavor perceptance?
Yeah. Hi, I mean, I have explained this thing. Silica, you have to just stir it. If you're, for example, you just take your flavor and putting in into the beaker or any bowl, you have to just stir with the handle. It will absorb all the things. So no need of putting into the other media and going with the other process and drying. So it is not something like that. In the silicon dioxide, the process is very simple. You are just putting the things into the beaker or whatever the mean you are having. So you can find free flowing powder but just by stirring all into the bowl. It is very simple.
Just I'm going to ask about what is the percentage the flavor changed the description about the action. I would like to ask it. Uh,
uh, yeah, you have asked the question.
my question is, if I'm using this mixture into the beverage or something else, or
Hmm.
if I make a use in encapsulated version, what is the difference between them? The flavor part. What is the acted in that flavor reactions? I asked it.
See, flavor is something, it is very relative. I mean, there is no.
For instance, for instance, flowers. I'm going to use this solution, and it gives some flavor. If I'm using in encapsulated versions, it's going to different flavor description. What makes it difference between them? I'm going to where I'm going to use this one and where I'm going to use encapsulated versions. It's easily to.
Yeah, see, silicon dioxide, it disperses immediately into the media. And if you talk about like encapsulation, if you talk about like cyclodextrin is encapsulated, your flavor is encapsulated in cyclodextrin. So if you talk about the percentage wise, it totally depends on flavor to flavor how it is that. And if you talk about how much flavor you are going to use, again, it depends which type of flavor, how flavors is harsh, what is a sweet flavor, what is a bitter flavor, which type of flavor it is. So it is not I mean, we can't say like there's a particular rule for that. It's again trial and error what you're going to take with the help of this. But yes, conversion wise, if your flavor is very thin like peppermint oil or less than that, your viscosity is very less, then your yielding will be around 75%. The thing is that uh, it is more economical and easy to handle. So this is one of the objective is that in other encapsulation process, you are going to have a big process of encapsulating your, maybe if it is bitter one, you're going to encapsulate it, that, and the process is bit longer. But totally depends on what is objective and what you're going to use, which type of flavor you're going to use. It is something like that. Hope I have cleared with you.
Thank you.
Guys, last question. We have 3 minutes left, so if anyone has anything on the mind,
Also in Emma, I
is
think
there an
some.
Emma? Yeah, I can see your hands raised.
All right, thank you, Dr. Ken. I just have one question.
Yeah.
The silica nanoparticles, uh, have you been able to look at forming a composite of it? Say you use cyclodextrin, maybe any of the kind of cyclodextrin, maybe beta cyclodextrin or alpha cyclodextrin, and maybe you use any other biopolymer like chitosan, and to see their improvement. It actually enhances therapeutic intervention, bioavailability, solubility of some very hard to dissolve API.
So you mean to combine cyclodextrins with chitosan or other polymers?
Yeah,
Yeah.
cyclodextrin and chitosan. Have you been able to form a composite of this silicon and check the pharmacological responses by using different API or any kind of API?
But with the
No,
silica or just the cyclodextrin and the chitosan?
Right now, that is cyclodextrin with the silicon, and that is cyclodextrin with the chitosan.
So with the cyclodextrin and the chitosan, there are results that so there are no products on the market, not even in later phase of clinical development or not even in clinical development. There is promising preclinical data, I would say, but nothing beyond that. For certain application, it looks good, but no one really took it further. As for silica and cyclodextrins, I don't really think that there are, but Nassim, maybe you know better.
no, no. I have not seen. I have not seen this type of formulation. I mean, commercially, it is. I have not seen. Maybe it is available. The world is very big, but I have not seen from my eyes.
Oh,
Okay.
okay. Okay, thank you. Because I'm looking at it as one of a special area of such formulation. Maybe there
Yeah,
could
if
be
if
improvement.
you can please
Yeah.
mail me, we can look into that and we can give you better clarifications on this.
Oh, that's okay. That's correct. Thank you so much.
Thank you.
Okay, so our time is up, guys, but thank you, Nazim, for the exciting talk. And I'm extremely happy that the discussion was so vivid and turbulent, and we could tackle a lot of questions. So I really appreciate the activity from everyone, and we'll come with a next masterclass very soon. Until then, take care of yourself. Have a good day. We see you next time. Thanks everyone
Okay.
for
Bye bye.
joining. Bye
Bye bye,
bye.
everyone. Bye bye. Take care.
Thank
Bye bye.
you. Bye bye, everyone.